Hi, I'm Josh. This is our gut microbiome project. Merry Christmas, in case you're watching this. Zach, shout out. Okay, the gut microbiome are the types and proportions of bacteria found in specific locations. The gut microbiome affects the health and diet of an organism. One important physiological impact is diabetes. Microbiome relates to diabetes because it affects how the metabolism works and how well sugars are absorbed. There is a direct correlation between the gut microbiome and diabetes. High fiber diets could prevent this disease. There was a study done where fecal matters of lean individuals to obese individuals. They found that increased their insulin resistance and was a causational relationship. Another important physiological impact is obesity. The gut, it is a causal relationship because many factors can contribute to obesity. The gut microbiome can still be the reason for some weight gain or weight loss. In a study by Kastner, they looked at a correlation of the microbiome and the BMI of individuals. The research found that obese subjects had a wider diversity in the gut than non-obese, as well as having a higher firmicute bacterial diets. The relationship is seen as correlational. So for our hypothesis that we developed was that the older dog will have a more diverse and complicated gut microbiome compared to the younger dog. This is because we believe that since the dog had more time to live, that they would have more time to accumulate different types of bacteria and it would be more diverse. What we studied in our experiment was 2 female flat-haired terriers. One was 6 months old and one was 4.5 years old. Both eat biscuits and table scraps. The younger one eats grass. The young one is 40 pounds while the other one is 50 pounds. So we started off by using a Qiagen kit developed by Qiagen Incorporated. It is designed for the isolation and purification of DNA from stool. We homogenized the stool in a buffer to prevent degradation. We then binded it to a filter and eluted it using a wash that weakened the bond with the filter. We followed the protocol exactly for this. Next, we used a Zemo kit developed by Zemo Incorporated. It is designed to concentrate the DNA sample tenfold. After the Qiagen kit bonded the DNA to a filter in the column, we washed it of impurities. We then eluted the sample at 1/10 of the volume. Thus, our DNA is now 10 times more. We followed the protocol for this as well. After this, we had to quantify it. We used this. We had to quantify the DNA, and we did this through spectrophotometry. We used a nanodrop. We used the nanodrop spectrophotometer to determine the concentration of our DNA and to estimate whether or not there is any contamination from proteins or RNA. We measured this in nanograms per microliter. How this works is spectrophotometer produces ultraviolet light. DNA absorbs light very well at 260 nanometers, which is also the UV range. Proteins absorb light at 250 nanometers, thus we can use the ratio of absorbance at 260 nanometers to 280 nanometers as a measure of the purity of our DNA. Pure DNA has a 260 to 280 ratio of approximately 1.5 to 2.2. Ratios below 1.5 indicate possible contamination with proteins. If the 260 to 200 ratio is far from the acceptable range, it means that our estimate of the DNA concentration may be inaccurate and the contaminating protein may interfere with later steps. After this, we did amplicon PCR. Now that we know the DNA concentration, we want to amplify the regions of the 16S ribosomal RNA genes, specifically the V3 and V4. This gene contains 9 variable regions interspersed between conserved regions. Sequencing these variable regions allow us to determine which types of bacteria are present in the gut. The expected size after amplicon PCR is 550 BP. Only our positive control yield the expected results because there was not enough expected DNA in our sample. This is because we wound up throwing out our negative DNA by accident. It didn't really work. For amplicon PCR, we had to do a thermal profile and cycle it. So we first denatured it by heating it to 95 degrees Celsius. Once the primer is bound, the temperature is raised to 72 degrees so that the DNA polymers combine, and we start synthesizing DNA. If you notice that we have an unusually high temperature, most organisms are denatured by temperatures much above 45 degrees Celsius, which is why we did this. And as you can see in our chart, we have the reasons why we did this. 95 degrees Celsius for the denaturing, and then we slowly lowered it down for annealing, synthesis, 72 degrees for spinal extensions, and storage of our products. We did 25 cycles. Gel electrophoresis. This is a technique in which molecules can migrate to a gel-like matrix so that they can be localized following electrophoresis. We use this to separate DNA, RNA, and proteins. In our experiment, we will be separating DNA fragments of different size using agarose gels at 250 volts. The purpose of gel electrophoresis is that it allows us to determine the size of the DNA fragments we are studying. In our experiment, we expect the size of the amplicon to be approximately 550 BP. This isn't exactly the case because we sort of messed up a little bit. We didn't have our negative controls, and our DNA was as pure as we thought it would be. Indexing PCR. For indexing PCR, we use the unique short set of tags to the ends of the amplicon so that we know what our samples are ours. We will use 2 primers, except only the 3 will attach. The 5 will contain the unique tags needed to identify. This allows us to identify and differentiate our group's samples after sequencing via those tags we put on it. And then after this, we will do PCR cleanup again. This is sort of similar to what we did before with amplicon PCR now, and the cycling we did earlier. The difference is that rather than 25 cycles, we only use 8 cycles. This is because we don't want to make large amounts of new amplification. So that's why we only did 8 cycles. Well, PCR cleanup now. We use the Adrian Core Ampere XP kit to clean up the amplicon from our amplification PCR action. The cleanup will remove components of the amplification PCR buffer, Mg2+, and DNTPs, and enzyme, as well as the unused forward and reverse primers. This is because they are unused primers and they may interfere with sequencing. We will do this once again later in the lab after the PCR. PCR cleanup. The principle of the amplifier kit is that the DNA will be bound to a solid surface. Impurities will be washed away, and finally the DNA will be eluted from the solid surface and collected. However, in our lab, the surface the DNA will be bound to small magnetic beads, and those beads only bind to double-stranded DNA of the amplicon, not the single-stranded DNA of the primers. DNA sequencing. We will sequence our DNA via Illumina misset. Indexing PCRs are denatured in this. The single-stranded DNA is attached to the surface of a chip. The chip binds millions of single-stranded DNA at different locations. Each strand is amplified so there are identical copies at every site. Fluorometric DNA quantification. We use this to determine the concentration of our amplified sample after PCR cleanup. Fluorometric spectrophotometer is accurate because, unlike UV, it can differentiate between DNA, RNA, and proteins. For our UV quantification, we had 3 negatives. We had 3 negatives, values 11.1, 2.2, and 2.3. They only are positive control yielding positive concentration, meaning their concentration of DNA is very low. Most of our 260-200 ratios are around 2.2 as stated earlier. You can see our UV spectrometry. None of our samples have bands except for the positive control, which is right here. The really dark ones is our positive control, right there. We only had DNA in the positive, so what we calculated, even though it was not enough, around 0.1 nanograms per milliliter. Our younger dog sample 37 had 267 reads, and our older dog at 308 and 18,227 reads. This is mainly because, well, we did throw out our negative control, so that was part of the error on our part, but, oh, we're gonna go over there now? Okay. Well, as we said, we originally did not do this. There was a mess up with the sequencing machine broke this year, so we wound up doing snakes. So this is last year's results from another group. So the 308, this is our older dog, sample 308. As you can see, there is 99.02% of bacteria. There's not much diversity in here. There's 0.98% other, which means everything in there is pretty much identifiable. Other just means that we have a small amount of bacteria or other things in there that just are quantifiable, or they're so small that it's not worth listing them all out. In our younger dog here, we have 75% bacteria and 24% unclassified. This is way more diverse than our older dog, which contracts our hypothesis that the older dog would have a more diverse microbiome. The unclassified just means that we may not know what they are, or we just don't know what they are. They could be something we haven't identified, or just something we don't know. But okay. So sample 308 is our older dog, and at the top order, the bacteriodales, makes up 43.5%. That's mainly what that is. And for our sample 307, you're good. You're good. So for sample 307, which is mainly clostridiols, and that's mainly found in younger dogs, so it helps with digestion. And 307, the top genus for the younger dog is mainly unclassified, which compared to the sample 308 for the older dog, which is mainly bacterioids. Again, this shows a stark contrast in what we thought. Clearly, the younger dog had a more diverse microbiome. The DNA extracted was unsuccessful. We had -2.2, -2.3, -13.1, and 11 nanograms per UL. What's UL? Okay, microliters. Yeah. So we got microliters. Negative values indicate DNA was low and had high contamination. In order to say that it has a high concentration of DNA, the 260-200 ratio of the results should be 1.5 to 2.2. Amplicon PCR had no bands except for our positive control, which had bands at 550 pp, so the positive had no cross-contamination. However, the absence of the bands at 550 and anywhere else means that we had insufficient metaDNA or high amounts of RNA contamination. We calculated our positive control to be 0.275 nanometers. This is not enough, as we would need 1,520 microliters to get the right concentration, so it was inadequate for sequencing. We lost our negative control, but we would expect a concentration of 0 because our negative control should have no DNA. From here on, we use previous data to examine the positive controls of the two dogs. This is because the sequencing machine broke this semester, so we had to rely on previous results from last year. That's basically it. Now our positive control led to 56,620 total reads with 100% passing the quality filtering. More than enough to get a good analysis. 99.98% in the same kingdom, with 82.63% being the same species of E. coli, 8%. Unclassified, 8% being Serratia, entomophilia, and less than 1% of other species. What? Okay, it's not 18? Okay. It signifies is that there is only about 25% contamination of other reactions in the sample. Reads from our sample. Sample 308, the older dog, had 20,034 reads for and our sample 307, our younger dog, 6-month-old dog, had 3,097 reads. Both are at least 1,000, so it is sufficient to base our analysis on. Sample 307. The younger dog, sample 307 through 8, kingdom level. The younger dog had the least diverse, with 100% of the DNA being bacteria, while the older dog had 0.04% other DNA. At the phylum level, there was major differences in the amount of firmicutes and bacteroids. The younger dog had 73% firmicutes and only 20% bacteroides, while the other had 34% firmicutes and 50% bacteroides. At the genus level, the similarities in Blatuvia and around 5%, bacteroids 18%, in older 11%, and younger in Prevotella 28%, and in older dog around 8% and younger. Now, according to the U.S. National Library of Medicine and National Health, an abundance of Prevotella is linked to the chronic inflammation in mucus and respiratory membrane, making it common in infections and diseases such as pneumonia. Now, the younger dog has a small amount in it, but in the older dog, it is the highest percentage possible, indicating the older dog is more at risk for lung infections. At the species level, they both have a large proportion of unclassified DNA, and other with unclassified at 28% in the young, and 31% in the old, and 25% other in the young, and 17% in the old. Other than that, the most prominent DNA in the young one was Collinsella intestinalis, and 17% in the old one. It was Prevotella colpri at 27%. Finally, the data we gathered from our experiment, it does not support our hypothesis. As seen here, the older dog had a way less diverse microbiome than the younger dog. We used the data at the genus level due to the majority of the species' DNA being unclassified or other. What we can be seeing is that the older dog only had 9 different genuses of DNA, the highest percentage being 28.65%, and the lowest 5.18%. The younger dog, by contrast, had 10 different genuses with a max percent at 18.18 and the lowest at 4.66%. This data showed that the younger dog had more strains and a larger range of the DNA in their microbiome than the older dog, making it more likely that the 6-month-old dog had a more diverse gut than the 4.5-year-old one. And that's a wrap. Merry Christmas, Zach. It was fun having you this semester. Thank you.